Thank you. Well, hi everybody. It's a pleasure being here, sharing with all you some of my research. So I'm been talking about the use of symbiotic microorganisms as a tool for recovering soil health in heavily polluted soils. Well, as we all know, soil has a regulatory potential, so it can act as a buffer of pollution. However, through the continuous and sustainable management and activities such as industrial or mining activities, humans, we provoke that in many occasions happens soil pollution. When these soil pollution events are strong, soil loses its role as a protective system of ecosystems, and there is the need to perform some ecological restoration. A big example in Europe of a heavy event of soil pollution happened in 1998 in southwest Spain when the Aznalcóllar mining spill occurred. It happened because the tailings dam of the mine broke down, causing a spill of over 4.5 million of cubic meters of acidic waters and toxic sludge. It affected a total of about 43 square kilometers, and there were taken immediate actions as the sludge removal and the top surface of the soil, as well as the large-scale addition of amendments. 5 years after this accident, the regional government declared all this affected area as a protected landscape to facilitate the remediation measures and to protect the area. And nowadays, the regional administration consider all this area as completely recovered. However, this is far from reality, since still 25 years after the accident, if we go for a walk in the area, we can find these bare soil patches, which are easily identifiable by the complete absence of vegetation. And there is present still high pollution, high levels of potentially toxic elements. And there were some studies performed that calculated that still 15 years after the accident, these soils represented about 7% of the total surface area of the first sector of pollution. Therefore, this is clear that there is still the need to work on the area, and we have a real scenario laboratory to carry out different remediation strategies. So in the work I'm presenting here today, we based on the concept of assisted natural remediation, which considers that the natural remediation processes of the degraded soil can be assisted or accelerated through the addition to the soil of different waste products or amendments which are economically and minimal invasive. In this way, we can use byproducts from the industry, and we can valorize these materials. One example of materials or byproducts from the industry that can be used as soil amendment is the dry olive residue, which is produced in large quantities, mostly in Southern Europe, where the olive oil industry is very important. However, this material has a high salinity, high content of phenols, so its application to the soil cannot be used directly in the soil. So one way to transform this residue is using saprobic fungi, which have the ability to degrade the phenolic compounds and phytotoxic elements of this residue. Also, in order to enhance the remediation process of the polluted soils, we can use bioremediation using arbuscula mycorrhizal fungi, which by processes of bioaugmentation in the soil of these organisms, they have the ability to immobilize or concentrate the toxic elements on their tissues by different direct and indirect mechanisms, which can be the production of chelating agents to the soil, production of glomalin, which is also immobilizing the elements, and it stabilizes the pollution in the soil, or directly retaining these elements in their structures. So with all this context, the aim of the study I'm presenting here was to evaluate the effectiveness of a bioremediation strategy for restoring the soil health in the soils polluted in the Mark Green corridor, based on the previous ideas that I talked about the valorization of human-derived waste and byproducts, combined with these soil beneficial microorganisms. So in this way, I'm going to try to schematically tell the main experimental design, which was based in greenhouse experiments using pots with this polluted soil from this area as a pilot scale application of these fungal inoculants. So we used in the pots, we used the wheat plants as model control species that it was grown in these soils with the different treatments for 45 days, about 6 weeks. For it, we used 2 different arbuscular mycorrhizal fungi species, the Rhizophagus irregularis, which was a controlled general species of fungi, and Rhizoglomus custos, which is a specific species of mycorrhiza, which was isolated from a soil naturally containing high concentration of these elements studied, the heavy metals and metalloids. Also, we used an inorganic amendment, which was a marble cutting and polishing residue, a powder, that gave us good results in previous studies in the field, as well as different organic amendments, which were vermicompost and 2 different biotransformed dry olive residues by 2 different saprobic fungi. So in general, the treatments established were all based in the polluted soil and marble sludge powder mixture, with the inoculation of the 2 fungi, the addition of each of the organic amendments, and the combination of the organic amendments with the inoculation of the mycorrhizal fungi. So as a general overview of the main results, at soil level, we could check that all treatments were very effective in neutralizing the pH from an original very acidic pH of the soil. It reduced the electrical conductivity, and in relation to the organic carbon and nutrients content, mainly the treatments based on the dry olive residue amendments were the most effective ones in increasing these parameters. Also, at soil level, we measured different enzymatic activities, and in all of them, we had the same patterns. Again, the dry olive residue amendments were the most effective ones, while vermicompost didn't perform that well. In relation to the parameters measured in the plants, after 45 days of growth, it highlighted the treatment consisting of the dry olive residue biotransformed by Coreopsis rigida with the inoculation of the arbuscular mycorrhizal fungi. And specifically, the Rhizoglomus custos, which was the specific one from the contaminated soil. Also, we measured the uptake of the potentially toxic elements in the plants, both in the left, in the roots, and the aerial part on the right. And as the plants didn't survive in the polluted soil, here we consider the marble treatment as to compare the rest of the treatments, which is like in the right side of the graphs. And it was mainly associated with the highest accumulation, both in roots and in aerial part, of elements such as lead, arsenic. Sorry? 5 seconds. Oh, okay. So yeah, we had the dry olive residue plus the inoculation of the arbuscular mycorrhizal fungi were the most effective treatment. So we see that the addition of these microorganisms they enhance the remediation process of this strategy for bioremediation. And yeah, okay, I was confused with the timer there. No. Well, I wanted to for 1 more minute about something else, but if there's. Well, I will just very briefly say that we kept on going with this bioremediation strategy with the conclusions from the greenhouse experiment. We brought it to the field in situ to this green corridor, And we saw that after 3 months and more after 15 months, there were interesting differences between the treatments, with different plant cover and species richness appearing, and the soil is developing in a promising way. So we consider that this application of the mycorrhizal fungi can be a promising bioremediation strategy for restoring this kind of soils polluted by the potentially toxic elements. And that's it.